I hope that you can hear me and see my screen. Yeah. Let me once again apologize for this inconvenience with the meeting settings. I don't know why Zoom decided not to let me to share the screen before I did the restart the app. So yeah, today is the second meeting of our second session of the Bioinformatics Club seminars. And I would like first to announce some schedule changes, which I think that you've already noticed, but still I think it's important to emphasize them. So today we'll be discussing calling of complex types of genetic variation, structural variants and copy number variants. And the meeting on somatic variation analysis, which was planned for today's meeting initially, will be on the next meeting on the 20th of May. And this was due to some of the speakers who will be actually, I hope, be in the leading role on that seminar, asked us to move it a little bit because of the overlap with the Orthodox holidays. And then we will proceed with the meeting, which we've skipped actually last time when we were scheduled to discuss genome-wide association studies. Again, due to some issues with speaker availability and overlaps with the conferences, we have to move it a little bit. So the current plan is to hold this meeting on the 10th of June. And we'll send out an official announcement of the final schedule as soon as we receive the confirmation of all of the people whom we'd like to see on these meeting. And as planned previously, we will end this session with the discussion of ancient DNA analysis, which for now is scheduled to the 24th of June. But again, we'll send out a formally finalized schedule after getting all of the necessary confirmations from all the people. Yeah, and as I've already mentioned, today we have the discussion of CNV and SV analysis. And today we'll be having more of a dialogue, I think, with German Demidov of the University of Tübingen. German, do you hear me?
Yes, we hear you well.
Great, thank you. Yeah. So German is a leading expert in this topic, so I hope that we'll be having a very nice conversation. Unfortunately, yeah, I have to say that the topic of structural variant analysis and copy number variant analysis seems to be less, I shall say, popular compared to short variant analysis, naturally, because well, any lab which is doing either practical or clinical cases or research in human genetics, I think almost all of them do short variant analysis for clinical or for research purposes. But with structural variants and other complex variants, it's, I think, not the case. Not any lab which does work in the field does these types of analysis for certain reasons. And I think one of the reasons is that, as can be understood from the name, yeah, these variants are just that, they're complex. And they are hard to call in many cases from next generation sequencing data. And there are also many challenges which are associated with the interpretation, which is also, I think, it's my personal opinion, less straightforward. And especially when we're talking about short read data. And yeah, that's my perspective on the reason why this is the case. But still, yeah, of the people whom I personally spoke to, I think, yeah, there are much more people who are involved in short variant analysis than analysis of CNVs and SVs, which is a pity, I shall say, because that's very interesting topic. Yeah, and we have some people in today's audience who have partial experience with the field, including myself, and I hope that we'll be able to ask for their opinion on some matters. But again, German today is our leading leading speaker, I shall say. Yeah, and before we proceed to our dialogue, I think I will take 10 minutes probably to introduce the topic for those in the audience who are less familiar with this matter, with the analysis of these types of variants. And then we'll move on to discussing the actual questions, which I think are important in this aspect. So first and foremost, I have to introduce the actual variant types that we are talking about. So last time when we were discussing short variants, we were mostly talking about either single nucleotide substitutions or short insertions and deletions on a scale of several nucleotides. Usually in the classifications which can be seen in different sources, we see that short variants are classified, let's say, under 50 or under 100 base pairs. So the thresholds may vary. And basically, yeah, we were also last time, I think all of the speakers pointed out that the task of short variant calling is performed relatively, or not relatively, generally well by most tools. And basically, we get 99 point something percent accuracy in most cases. And the issue is that as we increase the length of the variants, which has been noted, I think, for quite some time, even for variants which can be considered short variants in terms of they're still below this like 50 or 100 BP threshold. Still, as we increase the size of insertions and deletions, we get a certain decrease in performance of the variant callers. So as we increase the sizes of the variants, the task of identifying them confidently becomes more challenging, and naturally we can understand why it is so when we are talking about short read data, because short reads, yeah, typically for most commonly used platforms, they are maybe 150, 100 base pair long, and as the variant approaches in length to the length of the short read and becomes comparable at least, there are multiple issues which are associated with the alignment of such reads, which have large gaps or large insertions, and many other issues with parent calling subsequently. And as I've mentioned, yeah, this is true. The challenges arise even if we're talking about variants which are still considered generally as short variants. But the other topic of today's meeting is variants which are not generally considered short variants, which are considered structural variants, which involve larger variations, which are typically of greater length than these like 50, 100 base pairs. And these can be broadly classified into two principal types. This is not an exclusive classification, but still we have copy number variants, which includes naturally larger deletions and duplications, but it is not limited to, because of course we can have yeah, not duplications, but other copy number changes which are like giving a higher increase in the numbers of copies. But still, the idea of copy number variation is that we have an effect on the dosage of the gene or a particular segment, a particular region of the genome. Which again is typically several hundred, several thousand, or tens of thousands, hundreds of thousands base pair long. So a certain pretty large segment of the genome is either deleted completely from one or both chromosomes, or otherwise is duplicated or obtains or requires more than 3 copies. But still, yeah, here we are talking about imbalanced events where we have differences in copy number compared to normal. And another group is, of course, balanced chromosomal rearrangements, in which we have essentially the dosage of all genes of all sequences preserved, but the order of them changed due to inversions within a single chromosome or due to translocations between different chromosomes, whereby a portion of the sequence is moved from one chromosome to another, which disrupts very significantly gene regulation and may also damage the sequence of certain genes if the breakpoints of these variations are within the genes, within the gene boundaries. And of course, as I've said, this is not like an exclusive classification, because of course there are other types of imbalanced complex chromosomal rearrangements. For example, some translocations may be combined with not only the movement of one genomic region of one chromosomal part to another chromosome, but some portion of DNA can be deleted due to this event, or otherwise increase its copy number. So this, of course, can be there can be combinations, but generally, when we're talking about structural variants, we think there are copy number variants, and there are variants which are associated with rearrangement of the genome sequence, which is not, or at least not certainly associated with copy number changes. Yeah, that's just the overview of the subject in terms of which types of variants we are talking about. And next, we have to talk a little bit about the principal sources of information which are being used for the analysis of these types of variants. And here, there are essentially several approaches which are commonly used for identifying these types of variants. And the first and foremost, of course, there is the count or depth of reads that are aligned to particular places in the genome. And this is, of course, the methodology which is used for copy number variant analysis. And in this case, naturally, when we have a deletion of the certain portion of the genome sequence, we'll have a decrease of read depth in this region, or absence of reads aligned to this sequence. And otherwise, when we have duplications, we'll have an increase in the number of reads which are aligned to this location. And we can use encallers, the methods which are used for encalling will use the numbers of reads or the density of reads aligned to a particular segment of the genome to identify copy number variations. Another type of information which can be used in order to identify structural variations for short reads is the information about alignment of paired end reads, or the information on split read alignment. So basically, the idea here is that where we have certain types of complex genetic variation, either a single read can be aligned with a large gap, so like a certain portion of the read can be aligned to one location, and another part of the read will be aligned to a different location on the same chromosome, or even to some location on a different chromosome. And such split reads are indicative of breakpoints in the DNA, and can be utilized to detect structural variants. And similarly, when they have paired end information from paired reads, the idea will be basically similar in a way that the orientation of the reads in the pair will be also indicative of a structural variant. For example, if one read in the pair aligns to one location on the chromosome, and another read, the paired read, is aligned to a very different location on the chromosome, or even to a different chromosome, that's indicative of either a deletion or other kind of rearrangement on this chromosome, or involving several chromosomes. So this is the second type of information which is useful for structural variant identification. And the third category, I shall say, is the like methods which are based on de novo assembly, which I think are relatively less popular but still exist. Yeah, and these methods for structural variant analysis typically try to first take the reads and, not using any information about the reference genome, assemble them into larger contigs, and then use these contigs to align them onto the reference genome after the assembly, and hence as we are aligning larger sequences compared to short reads, will not have the problem of the length of the variant being comparable to the length of the read. And this way we'll be able to identify different types of variants by seeing that the certain large contig is aligned with a gap or with an insertion, or aligns to a couple of different chromosomes. So that's basically the idea that that's used by these kinds of methods. So these are the principal types of information which can be used for identifying different types of variants. And naturally, there are a plethora of solutions for calling structural variants and copy number variants. And at least again, this is my personal perspective on this matter, that compared to short variant calling, the different tools, despite using principally the same type of information, because despite methodological differences and difference in the algorithm, still the types of information will be basically the same for different methods. But the outcome is much more different compared to short variant analysis. So in terms of short variant analysis, usually when you compare two different variant callers, you'll see a very large degree of concordance between the resulting call sets, which basically is understandable because, as we've discussed, the task of short variant analysis is relatively straightforward. But with structural variants and copy number variants, again, despite certain information being available, still there is much more questions regarding how to handle this information, regarding the thresholds which are utilized to separate genuine variants from false positives, from alignment issues, etc. And as you can see from this plot, so this is a plot which was produced in one study, which is a 2019 study of benchmarking different structural variant callings in the niche communications. Here, there are the precision recall curves for the different tools, with dots indicating the certain default or otherwise preferred sets of settings. And you can see that actually the performance is first lower compared to what we expect from short variant callers. And in certain cases, we can see that the recall of the analysis in certain cases is very, very low. And then actually certain instruments, cannot at all detect even like 90% of true positive variants. So this is actually, I think, indicative of the problem that short read data are not that much suited for like a precise analysis of structural variation. So we can do this analysis, surely, but there are limitations of the technology which limit its application for comprehensive analysis of structural variants and copy number variants. And in order to have more comprehensive results, of course, long read sequencing are of importance. And as far as I see from the recent studies, there are more and more rapidly introduced into the practical applications. And with long read information, the performance is I think markedly better in many cases. But still, as far as I know, there are certain challenges even when long reads are considered. And the last thing I wanted to say before we proceed to the discussion of the matter is the specific, like zooming into the topic of copy number variation, because here we have the analysis which is focused on different types of structural variants. But generally, as I mentioned, there is a specific category of which are copy number variants, which are usually detected using specifically read depth or information. And the calling of these variants is also challenging, but in many aspects, it's more straightforward. And still, it requires certain specific data sets to be involved. For example, when we're talking about copy number variant discovery, there are two principal types of methods which can be used for the CNV analysis, which are the so-called reference-free methods, which will use information about read depth across the genome. And typically, this it is possible to use these methods to analyze whole genome sequencing data when you expect the coverage across the genome to be uniform, and of course, correcting for accessibility of different regions for short read data. You can basically identify places where the coverage is systematically lower compared to the genomic baseline, or systematically higher compared to the genomic baseline, and thus designate these areas as segments with copy number variation. But as I've mentioned, this is specifically applicable for full genome sequencing data, because when you have targeted sequencing data sets such as exome data or panels, naturally you don't have enough information, because you have gaps in the coverage, which are simply due to certain regions being targeted and certain not being targeted by the kit you're using. And secondly, there is much more variation in coverage across different regions in the same sample in targeted sequencing. So if you take a typical exome sample, you'll see large variation in coverage of different exons without any kind of copy number variation due to efficiency of target capture mostly, or PCR amplification if you're using amplification-based methods. And this, of course, hinders and prevents the application of single sample reference-free methods. So if we are using targeted sequencing data like whole exome or gene panels, we'll have to assemble a reference set of samples, and the quality of this reference set of samples will actually be pretty important for obtaining accurate results. So the figure on the right, which is taken from a nice review by Kristen Gillison's group in the Netherlands, shows the results of copy number variant analysis, where each dot is a segment of the chromosome, and the position of the dot on the vertical axis corresponds to the estimated copy number. And here, this is the same sample, which is shown on the two panels on the top and the bottom. But basically, the reference set of samples used for the analysis is different. And you can see that there is large differences in the estimated copy number of multiple segments when they're using different sets of samples as reference when running the comparison for copy number variant identification. So that's another important aspect that CNV analysis, though in certain ways more straightforward, has these issues, especially for targeted data. These things have to be, of course, taken into account. So that's it for the parts of the problem which I wanted to talk about in this introduction, just to give a brief overview of what the field is like and what are the main issues here. And with that, I think we can proceed to discussion and listening to German, who I hope will share his practical experience with this matter. And first and foremost, I'd like to ask German, so could you please tell us about the CNV and SV analysis itself? So for you, is this a part of the analysis which is routinely performed for all samples, or are there any like specific aspects of like when you for example, obtain no results of short variant analysis and then the sample goes for CNV and SV, or like how is it organized in your case?
Hello, everyone. I'm sorry for being invited expert one more time, but this time I'm actually expert in the topic. This time I can actually speak kind of being opinionated about the topic. Okay, so of course it's both CNV analysis and structural analysis are the parts of standard analysis nowadays. What I'm probably especially proud of that since 2018 in our clinic and in like 5 different other clinics in Germany, it's my tool that I developed is used for the CNV analysis there. It's not always possible. If you don't have enough samples for a big control cohort, for example, some collaborator sends you, I don't know, 10 samples with exome sequencing results, and then you don't have enough samples to normalize between them. You kind of just say, well, we don't really do this, especially if the cohort is, for example, sex biased, only females, for example. It becomes really difficult in this case. But it's a standard part. Just you need to be careful. You need to reject the results which don't look good for you. You don't need to waste the precious time of the clinicians who have to interpret these variants if the results are not good. So you have to be honest in this case. You try to analyze everything, but you have to be careful and filter out a lot of results after. That's summary of what I want to say.
Well, yeah, I think that's reminds us of the discussion we had last time, because yeah, that's pretty much the same logic about the filtering of candidates.
Here is even more important. So like it's not even the quality control of variants, it's quality control of samples too. So if the sample doesn't look good, you don't trust anything in the sample, you throw it out.
Okay, so you've already touched upon this matter, so I think that we can talk about it in more detail. So when you're talking about the reference set of samples, yeah. So what is your like attitude towards the construction of this reference set of control samples? What other features do you take into account, and do you like maintain a single like gold standard set, of do you make adjustments for different samples? What's your practical approach?
It's a good question. I started my way into bioinformatics with developing of the tool for CNV detection in 2014. And my PhD was about the developing of tool for CNV detection. So I tried a lot of ways to normalize, to choose the reference sample, the reference cohort, and there are different approaches. For example, in Nijmegen, this Christian Gillissen group, you showed the result. It's the biggest medical genetics center in Europe. They use the last 300 or around samples as the control cohort, because the wet lab is changing and you kind of wet lab affecting the quality of like the uniformity of coverage, the similarity of coverage between different samples. So you better rely on the samples you sequenced recently, because they may be very different from what you sequenced several years ago. In our clinic, it's a bit different. What we did, we have I don't know, maybe like 20,000 exomes currently, and I don't know, 10,000 genomes. And what we do, we calculate coverage profile for each of the samples, and we store a huge matrix of correlations of different coverage profiles. So we select, I don't know, 150 or 200 samples as a control cohort for the CNV analysis, using this huge correlation matrix. And each time we process a new sample, we add it to this correlation matrix. Just choose the most similar ones, no matter if they're the latest ones or they were sequenced 5 years ago. Maybe you returned, someone submitted you the older version of the kit, something like this. So we use bit different approaches, but the idea is the same. The samples have to be as similar to each other as possible for the successful CNV analysis. That's mostly it.
Is there any like attention you pay to the phenotypes of the samples? Yeah. So is it strictly like controls which do not have any like causal CNVs or like do you consider anything about the phenotype of the patient before adding it to this control panel?
Overall, it depends on the approach in the clinic, but in a rare disease, if your clinic is not specialized in some particular like breast cancer, where, for example, you may see deletions of BRCA1 or BRCA2 quite often, and there you kind of lose the power of detection there. If you have a general genetic clinic, which analyzes many different diseases, not specialized in a single one, we kind of can rely that CNVs are relatively rare. And if you use correct statistical methods when you develop your tool, which are not sensitive to outliers, like the standard average, if you use the standard average for normalization, it can be heavily affected by one outlier, by one duplication or one deletion. And if you use the robust outlier statistical methods, then you can say like a couple of the CNVs in my cohort of several hundred samples don't play any role. There are some regions which are normally copy number variable in human genome. So in this region, this idea would not work. Just choosing like median instead of average would not work at all. Yeah, overall, you rely on the fact that CNVs are rare and they are not recurrent in rare disease. Cancer is a bit different approach, of course, but in cancer, you often sequence normal and tumor there, but we speak about germline, and then you use just the other affected cases as the control cohort. You don't really select them by a phenotype. What can affect is the large family. If the large family is in the analysis and they sequence within the same batch, it's quite probable that the allele frequency within this batch will be relatively high, and then you may filter it out. So in this case, you have to be careful. Large family means modification to the filtering there. That's most of it.
Okay, yeah, thank you. That's that's an interesting about the large family. That's I think an interesting argument. Yeah, okay. So I think that you've already let's say mentioned that you have developed your own solution for CNV analysis, but still I will ask you about your choice of methods because besides CNV analysis, there are the general structural variant analysis for the balanced rearrangements and other types. Yeah, so what's your choice of instruments for to these tasks? And I understand that you about your custom CV approach, but in general, what's your maybe opinion on other commonly used tools? Whether you have maybe some criticism of the approach, yeah, anything you'd like to comment on.
Yeah, the whole field of detection of CNVs, it started even before the NGS started. It started with array, and then methods, algorithms were developed 20, even 30 maybe years ago. So what I definitely advise against is using the tools from 2011 or something. And the picture you showed, actually, it's the Kitchen Gleason group. They're actually using Conifer tool from 2011, actually. And it's bad, it's terrible, but since so many samples are processed with this tool, I think they just they don't want to change the tool. So my advice, just if you start in 2024 the analysis, don't use the tools from 15 years ago. It just like bury them and never. My tool is not much more modern, but it's like it's not 15 years old. Currently, for rich people, the standard more or less is the same. It's just take Dragon and run Dragon. But for people who are not that rich, there is a set of tools which are quite good. Everyone uses Manta for structure variant detection in both exomes and genomes. Yeah, we can talk about structure variant detection exomes, even though naturally you wouldn't expect a good result from it, but it's still possible. So people use Manta even though it's not the best tool on the market. It's just provided by Illumina. It was introduced in 2016 or 19 or something like this. So it's
I think
really
16.
old, yeah, and the results are not good, but people use it, and we also use it. Just again the backward compatibility. Otherwise, we have to reanalyze the 30,000 of samples. It's not bad, but there are better tools here. And in CNV detection, again, exomes and genomes, they use different tools normally there. And yeah, you normally choose the 2 or 3 tools to merge them. One should be based on the read depth, one should be based on paired end information, and one tricky with the split reads, but they are like in specific situations, and the variants which require split reads are rare. It's, for example, mobile element insertion or the tricky genes there. Also possible, but it's rare. So if you don't want to spend your time on this, use paired end and read depth based tools. Yes, Manos?
Okay, and yeah, as you've mentioned, they're using different tools. So let me follow up with the question of because there are methods which are like the ensemble calling methods. I think Parliament too, I think, and others which I've seen. Of course, not having too much of a practical experience with this. So basically, what's the approach here? Is it using these sort of certain methods which are like ensemble in nature, or just running two different calls in parallel and do some sort of custom merging on some stages? What's the best way to do it?
In my opinion, CNV detection, especially read depth based CNV detection, is very noisy. And merging results from different colors, you just merge trash from all these colors. Let you deal with errors of one of them. You don't need to deal with errors from all of them. Just to the best one. Normally, I'm very timid about my own results, and I'm very modest. But in this case, I can see my tool actually works really well. So this particular one I can recommend to use. Some of the other tools I know that may be comparable results, but with the good hands, you can make my tool working really well. And we use just this one and Manta in pair and my tool. It's used even for long reads because the aneuploidies and long reads definitely. If you don't catch the breakpoints, you don't see that the aneuploidy happened with the basic colors. So you need to do some sort of read depth analysis anyway in Nanopore or PacBio. So I would recommend to choose the best tools and don't multiply errors from many of them.
Okay, yeah, I think you've mentioned your tool multiple times, but yeah, sorry for me asking this, but still, like, can you give the name of the tool? Is it like freely available or not?
Yeah, absolutely. It's available. It's called Clean CNV, and it's the
Ah.
code is horrible. I'm sorry for this, but it works. It works really well. If you manage to start it, and people are complaining there is no container, and there will be no container because like real mathematicians don't use containers. No, I'm joking. I'm just too lazy to make container. It's implemented as part of the clinical pipeline, so it's also open source and free. Our clinical pipeline. So if you manage to install this Clean CNV will just run out of the box. So from the clinics with whom we work, there is no requirement to make a container for this tool. That's why they use this big pipeline that we have, and everything works for them out of the box. That's why I'm not. If someone wants to make a container for this, I would mention you everywhere possible. Yes.
That's a nice opportunity. I think we should look into it. Yeah, it's nice because actually, yeah, I've heard people using clean CNV, yeah, and I think we've already also cited it once. Yeah, and I didn't realize that that's actually you behind it somehow. I don't know. Yeah, nice. It's still to mention that. Yeah, I've heard much of the positive feedback about it from different people. I have to say. Yeah, you've mentioned like right here the long reads, yeah, the nanopore PacBio, and I think it's also important here to follow this up with the question of the usage of long reads. So is that a standard practice again for in your center and other centers you like have connections with? And if not, so what are maybe the principles of when these long read sequencing is applied? instead of, or in addition to short reads?
Unfortunately, long reads started to use more and more, and then the expert in short reads like me become less and less valuable. So previously, everyone was like really impressed when I look at the coverage profiles, like this is false positive. And it was really cool, the thing to do. But with long reads, the whole art part of the CNV detection disappears. Unfortunately, if you have reads long enough, you can detect whatever you want, translocations, inversions, so it becomes really boring. And it's more and more difficult to impress clinicians now in Europe nowadays with the different CNVs that you detect. As this, you really need to look some interesting examples. I don't know, 10 different SVs in a row. Like this one are still impressive, but it works really well, and it's a standard part. The only thing what I can just say, don't try to save money on coverage. When people try to save money on sequence with 10x, I know, PacBio, Hi-Fi, or Nanopore, yeah, it's cheap, but it means that your sensitivity drops quite a lot. So better don't do it at all than do it with 10x, to be honest. Yeah, so 20x or something, then it will be better. And you lose a lot when you try to save money on the long reads. Yeah, the prices are improving, and now there are new methods which involve indeed assembly, as you mentioned, which was previously impossible to use with short reads. Like you won't do de novo assembly of human genome, but nowadays it's a fashionable thing, to be honest. You just do the novo assembly of the genome and compare it with the reference genome. So here, all the structural variants are just like from assembly. They come from assembly. It's a pretty accurate thing. Yeah, so yeah, unfortunately, the art of CNV detection is going to disappear because of long reads. Yes.
Yeah, so maybe I should like once again ask you if you can comment on are there any like principles when certain samples are sequenced with long reads in your center or not, or like this is just a question of like whenever it's available. Well, are there any like rules, let's say? So, for example, if we didn't see anything pathogenic with short reads, will repeat this with long, or are there any like roadmaps, workflows for choosing like to sequence the sample with long reads, or is it?
The approach is different based on this scientific center, of course, but indeed, currently there is at least move. Is definitely a step which already happened. Is the no one is doing exomes anymore. So exomes, like, they still exist, they still process, but there's a whole change in paradigm. It's genome first. So people don't wait negative exome when they proceed with the short read genome. And with long reads, currently we know that most of the variants are short variants, most of the disease-causing variants. And with nanopore, with 30x standard coverage, you still struggle a bit with more than from Illumina. And thus, short read sequencing is not dead in this sense. But if you expect, for example, repeat expansion to be the cause for your disease, it's possible because your phenotype can be very bright there, and you can see that exactly this particular gene. Yeah, you can do PCR, standard fragment-based PCR, and you can run the nanopore or long reads out of the box immediately. But indeed, I would say the current standard is to wait until negative Illumina whole genome. Illumina, BGI, whatever people use. Yeah, so it's still the second step in the protocol.
And in terms of Hi-Fi versus Nanopore, you mentioned both. Like, which is the preferred option for like at least in your practice?
I think it all depends on which contracts your scientific center have. If you're friends with PacBio or you're friends with Nanopore, and it's very expensive to be friends with PacBio because their sequencers are really pricey. So yeah, from what I know, it's purely based on like which sequencer you have in your building. In our building, we have SQL somehow, SQL 2 probably, but we have so much more Nanopore, so we almost don't use PacBio, at least for humans. So more like diagnostic solutions. Nanopore is not validated for diagnostics yet, but mostly diagnostic negative short-read genomes go through Nanopore, of course. Here, but yeah, it's a question of taste, I would say.
Okay, yeah, because I think that in terms of this, that was my understanding of the matter is that if we are talking about moving to like long read genome first, probably it should be PacBio versus Nanopore, maybe. I don't know, because at least my impression is that Nanopore still has these issues with reproducible biases, and hence you have more issues with calling short variants from Nanopore. Is that true, or am I missing something?
Indels are problems. PacBio also suffers from indels there, but PacBio costs a lot. Like if you have money, then yeah, go with PacBio. But it's very pricey.
Yeah, that's definitely true. And I think yeah. Um I have seen very few people who have access to PacBio. Yeah, and much more people who do something with the nanopore. Probably this somewhat connected to the issue of price. Yeah, that's nice. Yeah, I have to also maybe to comment a little bit on what you mentioned about the like de novo assembly and the short reads versus long reads. Because yeah, once we've had tried running, there was a tool which I think in some time ago has been quite like mentioned several times. It was called Fermi-Kit, produced by Heng Li, who is naturally the author of SAMTools and other great tools. Yeah, and we've tried to do this with, like to try to run certain analysis with short read data, yeah, and the results were absolutely like messy. So that was the very first attempts to do it, and it was yeah, very bad. So after that, we've switched to other tools for the specifically like limiting ourselves to CNV analysis and using specific tools for deletions and insertions. Yeah, that's I think an important thing to mention regarding short reads. Uh yeah, thank you. That's very important. So now I think we can proceed from the general issues of calling to like the subsequent steps, the same way we've done for short variants. Yeah, because yeah, when we were discussing the short variants, we were I think talking a lot about the filtering, etc., the strategies for that. And yeah, discerning and like this discriminating between true variants and non-true variants. And again, last time when we were discussing short variants, we were mostly talking about keep everything and let the interpreter like do the job by maybe like stratifying the data set by quality. So in case of like CNVs and SVs, again, my impression is that here the filtering can be more tricky because yeah, as you mentioned, yeah, the results in some cases can be quite noisy. So maybe other like any specific strategies, maybe some thresholds which you like employ standardly. So what's your way of dealing with the raw results of calling?
I would say the first thing you need to do is to check the results if you have too many CNVs. And if you have too many CNVs, it's because you failed the result, and you may try to do whatever filtering you want, but the CNVs calling won't be reliable unless the CNV is huge and there is a type of signal which people don't usually mention in the papers. So you were showing the types of signals which is used, but BLE frequency becomes really a valuable source of signal there. So you can see traces of duplications or deletions in data which is actually the coverage is super noisy, but the BLE frequency shows the nice like. But this variance should be huge, like megabases of genetic material. So first thing first is to do QC control. If the sample is bad, you may try to find another control cohort. You may try to normalize it differently, but most likely, sometimes you use different tool, and with different tool, it may give you better result. But overall, I would say if you do it in the bulk, if you do it in the part of routine clinical practice, you just give up on the sample and say like CNV analysis wasn't possible. It's unfortunately it's unavoidable. Like 5%, even in the like with all the wet lab precautions, 5% of the whole exome samples probably won't be really suitable for the analysis. And then Yep, and then the same strategy as before. So as with short variants is you're reducing the quality filters. So in the beginning, the clinician, they get only the very good candidates, very strong, technically strong candidates. But most of the time, they show nothing. And it was the request of clinicians to make this more relaxing filtering, because they normally have several hours per case, and you normally when you work as this genetic interpreter, genetic counselor, you're really eager to solve the case. And if you just see only strong candidates, you miss a lot. So they insisted for us to include this stepwise reducing of quality. So in the end, if you go to the lowest part, you will see a lot of like CNVs, where like 99% of the CNVs will be false positives. But still, they hope to find some match between phenotype, something like some additional source with like family information, where you can actually solve the case. So it's much more tricky than with short variants, the filtering. But the main principle is the same. You start with the good candidates, you find nothing, you move to the worst candidates, and then to the trash in the end, just to be sure that you did everything for this particular patient to try to find the diagnosis.
Excuse my ignorance in this matter, but in terms of the actual stratifying the data in terms of quality, so what are the metrics which are applicable to CNVs and maybe other types of structural variants? Because with short variants, I think it's more straightforward. You naturally have in the VCF files, you have like quality scores or genotype qualities, and then this. At least this for the simple kind of approach, it's like easier to do it. But what in my opinion, but yeah, what are the standard metrics like scores which should be taken into account when sorting the prioritizing the candidates in terms of quality?
When you perform the analysis of rare CNVs, and most of the clinical analysis is analysis for rare CNVs, and also multi-allelic CNVs is a different story. So you assume that all your control cohort is deployed, and only one or couple of samples are not deployed. So what can you use is like, what is the how dissimilar is to the deployed to expected deployed value, the coverage, for example. So you may use p-value, for example. You may make a t-test, and compare like this particular segment has higher coverage than everything else. And there you naturally get a Wilcoxon test, whatever, just the p-value. But I personally dislike this look for outliers, because germline copy number variants, they are not just increase of coverage or decrease of coverage. They're very specific increase of coverage or decrease. So it's like 2 copies become 1 copy. You expect the coverage, read coverage, drop to 50%. You don't expect it to be 70% or 80%, or just like wavy pattern in the middle, like at some positions 20%, some positions 80%. No, everything should be like along the straight line, and 50%. It complicates the detection of mosaic variants, of course. But if you look exactly precisely for the germline, then the bias factor or log likelihood of the comparison to two models. One model is duplication, for example, or deletion, and the second model is deployed. You compare them, divide one with another, and take log likelihood, and then this. Secondary metric is the allele frequency, of course. It's super important there, and other different, like uniformity of coverage inside the CNV. You may use something like this. So yeah, all these metrics are different from different exome enrichment kits. For example, all the exome enrichment kit has super high variability of coverage, and there you cannot really get detection of small CNVs of 1 exon, 2 exons, and nowadays they're much better. So this quality filtering for genomes is more uniform, but for exomes, it's very different there. Also, we should remember that our reference genome is European centric. So the African or Asian patients will have more CNVs than the European. And if you use just number of CNVs as the quality score, then you may just filter out good quality samples of different ethnicity here. So this is another thing you need to keep in mind. Yeah.
Okay, yeah, that's I wanted to, yeah, because you're talking about the models compared like to duplications and deletions, and my hypothesis is one to miss cases where not mosaic there, but still cases when you have more than one additional copy, like say four, if you have a total of 4 copies or 5 copies, or like do you test for all possible like non-fractional copy numbers, or what's the approach here?
Different approaches. For example, in somatic, you may actually, because you have to take into account not only clonality, sorry, not only the copy number, but also the clonality, the heterogeneity of the tumor, and the purity of the tumor sample. So there you cannot really check all the possibilities, even though my tool does this. But it goes up to like copy number 20 or something like this. After copy number 20, it says like whatever, it's just a lot here. You basically check indeed. In my tool, you check like copy number 0, 1, 2, 3, up to 8, I think. And there it basically cuts the coverage there. So if it's copy number 10, it will still think that it's copy number 8. It will provide you just copy number 8. Other tools, for example, they just provide you p-value. It's very different from the deployed. Let me say this. And then it's up to you. You have to genotype after. So they divide two steps. First, discovery of the variant, and then genotyping after, like this. So depends on the developer. I made this choice, and it works.
Yeah, it's nice that it's you don't only like check for like 3 copies or 1 copy. Yeah, that's it does it up to 8, as you say. Yeah, that's basically what I answered. Yeah, any like increases in copies which are not non-fractional, not in a sense. Yeah, and I think, yeah, here you've mentioned yeah, a little while ago the low frequency issue. So, and this of course, yeah, if you're talking about the low frequency, you have to use some sort of reference, yeah, to have this low frequency information. And yeah, here I need to remember, yeah, I think it was a ESAG in the COVID times, probably 2020. There were 2 talks on sets of SVs. There's not for CNV specifically, but I think for generally for SVs from 2 groups. One was Nomad, and another, I don't remember the name of the project, but basically I think IBRE Hall was one of the people in there. And they've mentioned the comparison of the sets of SVs which were produced for like comparably large cohorts in these 2 projects. They've they've tested the overlap between these 2 studies in variants which have a frequency higher than 1%, which you expect to in a large like sample of 10,000 people. You expect this to have a pretty high overlap, and they've seen, I think, an overlap of around 20% between the 2 data sets, which were both of similar ancestries. So that was indicative of the kind of like noise and problems with calling these types of variants from short-read data. And yeah, so this is naturally raises the question of how to choose the reference data set, like the reliability of the data sets, and yeah, how to employ these external resources in your like filtering of candidates. So what can you say about this part of the analysis?
Yeah, it's pretty expected to have this 20%. CNV detection, as I said, is still in quite a terrible state. Maybe with long reads we'll do better. But basically, when you perform CNV calling, normally you expect everything to be deployed, and then you look if something is 50% more, then it's like duplication, 50% less deletion. But there are pieces of human genome which are normally like copy number 8 in human genome or copy number 10. And the copy number variants, they're like 10, 9, 8, 11, 12. And if you assume that majority is deployed while it's actually 10, you don't expect the coverage decrease of 50% or increase 50%. It increases by 10%, like each copy number step. So there, the classical algorithms, they definitely fail in this case. So you have to run the special calling. In my tool, it's realized as the mixture normals. You don't say that majority is deployed. You just make like Gaussian mixture there. And thus, if you don't apply this, if you just rely on the number of deletions called in this particular region, you may be terribly wrong. If, for example, if 50% of your samples, let's not even go to this copy number 10 complex cases. There are regions where 50% of human population have copy number 1, for example. And then you think that this is diploid because you say like majority of my samples will have diploid copy number, and then you have everything like copy number 4 or copy number 0 for you. So it's always related to the baseline here, and it depends how you make it. You also have to keep in mind that when we perform genome copy number changes, we normally segment genomes into windows, like 1 kb windows, classical thing. But sometimes in human genome, there are elements like retroduplications, which are like mRNA ancient copy inserted somewhere in the genome. So there, this window basically will cover this mRNA and you will see nothing. But if you work with the exome data, with the target data, you will see the copy number change there, copy number 2, 3, like it's retroduplication is actually creating the copy number there. So these things you have to look at with a different scale here. So there are tricks, and low overlap is not surprising.
Yeah, that is I think it's the rule is not surprising. Yeah, but yeah, maybe I missed that in your answer in terms of the specific resources. Yeah, like if there are any like which can be like obtained and used, let's say of like annotation filtering pipeline. What are the data sets which are like the proper ones to extract this kind of information?
There was a good data set from 1,000 genomes, but it was very old, and the resolution of the variants there is quite big. So it's like events which are 5 kb and duplications 5 kb, deletions probably smaller there because you can use per dended information there. This one, there is a resource called DGV there. It's also, but this one I personally don't really like because I think it's full of trashy submissions. People submit the low quality results as the real variants there. GNOMAD, I think it's good, but I didn't go into details. I think they use just JTK MCV routine there. I'm not exactly sure, but I think they don't really detect this polymorphic sites, multi-allelic CNVs. So in these regions, it may fail you. But overall, in multi-allelic sites, you have no power there unless you're really extremely smart and can deduce these models in your head. So you can simply ignore them. You can remove them from your results and say that everything which is in these difficult regions, I don't look there, even though it may be a gene like SMA gene, spinal muscular atrophy. It has perfect parallel there. SMA1, SMA2, and then many people simply ignore looking there because with short reads it's impossible to distinguish them. It's possible, yes, it's possible. There is a mode, SNV, which distinguished to 1 and 2nd, but there are many other genes like this, like SMA, and they not necessarily have this SNP. So you need to use the databases for annotation, but better you perform your own database from your cohort and your caller, because your CNV caller will have your artifacts specific. And then if these artifacts are frequent, you'll be able to catch them. And you won't be able to catch them if you just download GNOME at frequency there. And this, I also recommend to run once, if you have 1,000 genomes or 1,000 exomes, you can run once this multi-allelic calling there. So you can find in your data which are the distribution of the multi-allelic sites. And otherwise, yeah, just annotate with everything and be careful about the difficult regions, even though they may be medically interesting. There may be really causal genes there, you still really cannot overpower the short reads there. Sadly.
And in terms of difficult regions, I think that there are different tracks, so like bed files which can be used for this purpose. Like, is there any like specific? Do you have any specific like track which you use, or do you use it in general? Because I think that there are
Yes.
some like accessible region beds from different sources. Yeah, do you have?
We have our house. It's again, it's open source, so we have the resource that we use ourselves. The multi-helix CNVs with allele frequency more than 2% are annotated there, and everything which is below there, you most of the time you can rely on the deployed. That most majority of the samples are deployed. So yeah, I can share the link.
Okay. Yeah, thank you. Yeah, I think we can with this we can segue into the annotation problem, which we've already started discussing. I think, yeah, with the discussion of allele frequency. Yeah, but still, as again in terms of short variants, and this is again my lack of experience, like practical experience with copy number variant analysis, for example, in particular. I know that standard annotation tools such as, for example, web, yeah, if you use Manta, let's say, yeah, you get a standard VCF with the annotated, with the structural variants, and basically, yeah, I think web does, at least to some degree, annotate this, as far as I remember. Again, excuse my limited experience with that. In short variants, of course, there are just typical standard annotation pipelines, again, web, SNPF, and OR. And in terms of these more complex types of variants, so the first big question which I have is that, well, what are the like preferred annotation strategies, and what kinds of information is important? Because I assume that that has to do, of course, naturally with the genes that are affected. Yeah, in terms of the genes within the CNV, then especially probably for SVs, for example, the ones that are at the boundaries, the breakpoint location. So, like, what are the important features that have to be taken into account for both CNVs and like other kinds of structural variants, like inversions, translocations, etc.
Now there is more or less a standard tool for the annotation of this variance. It's called unknot SV, and everyone uses this tool and provides you, I don't know, 100 columns of information which no person can in reasonable time comprehend. So you really need to keep only like 5 of them if you really want to go through the list of variants in your particular patient there. And you need to keep in mind the type of the structural variant and the fact that structural variant callers, they don't often annotate the structural variant. They often just write B and D, which is like breakpoint, simple. It may be translocation, it may be something there, it may be just duplication. And with your eyes, you see that it's duplication, but the tool is not smart as your eyes. And the tool just says like, it's just breakpoints, just whatever. So the most important annotation is the strategy that we use in the huge reanalysis project. We analyzed 10,000, like 20,000 exomes and 3,000 genomes from unsolved patients. Is visualization. So you try to put everything not in the Excel table, but as the tracks in IGV. So load the coverage profile, load the allele frequency, BAM file, breakpoints, like, and take screen. You may automatically take screenshots of the left breakpoint, right breakpoint, so you can comprehend. Our brains are much better working not with the Excel files, but with the like with, it looks a bit like a cockpit of the fighter jet, so it's so complicated, many tracks, many plus, but at some point you get used to this and you spend like less than a second to reject variant if it's not good for you. So the Excel tables, of course, they also work there. What's important, yeah, it's there. If it's a deletion somewhere in the middle of the gene, normally it's the and the mechanism of action of this gene is like loss of function, then you can easily solve this case. But if it's duplication, it becomes tricky, and then you need to look for the papers. If it's intragenic duplication, then it's kind of okay, because you may assume that the the gene in this configuration cannot really work. But if it's the duplication of the whole gene or part of the gene, then only RNA-seq can actually show. And what's super weird, recently we discovered that it causes quite a lot of fusion genes. and how the duplication can cause fusion. I really don't know. Ton of duplication, so like one part inserts after another, shouldn't be. But in with the duplications, you need to do literature search, and probably you even have to do RNA-seq. Deletions are much easier. And the other types of structural variants, it depends if it breaks the gene, like translocation breaks the gene. Then you may assume that this particular, if it's this gene is from OMIM and it's known, and the phenotype of patient is matching, inheritance is matching, then you may assume it's okay. But if translocation somewhere, I don't know, 10 kb from the gene, does it break the regulatory part? You don't know. RNA-seq can show it only this. Yeah. So my summary out of this is just visualize. Try to visualize everything, and with visualization you will be better than with Excel tables. Whatever those Excel columns you will make, you will get lost there. Try to visualize.
Yeah, and speaking of visualization, I think let me check. Do we have Gennady Zaharov here with us? I think we have. Yeah, Gennady, are you with us?
Yeah, I'm here. Just usually it takes me quite a long to unmute my microphone.
Yeah, thank you.
That's okay.
Thank you. Yeah, because why I asked because I had to introduce the Gennady, who was the permanent participant of the first session of our seminars. Actually was involved in a project of developing a genome browser back at EPAM Systems, which was concerned in part with visualization of structural variants. And Jan, if you maybe can say a few words about what types of information have you tried to represent and visualize for these structural variants? Yeah, what was your strategy?
Yeah. So what I did, in fact, what was our strategy? Just to note that when this project has started, I had quite a small background in bioinformatics. And so but in the same time, we needed to prove our capabilities to develop something for the life science. So we developed a new browser because it is quite obvious thing, it's visualization. And one more thing that developing some new calling or annotation algorithm was it's a different and complicated thing with you need a lot of scientific knowledge to do this. And we tried to develop some quite simple and robust approach. So in fact, this is not only the visualization of the structural variations. I can show you, probably I can provide the link to the genome browser. It's not actively maintained, but it's still alive and still capable to work. And what we in fact did, we created, we understand that most part of our customers use TightGV. And TightGV, it's very good when you look at the alignment, at the bump files. But in fact, the visualization of variations in IGV, I don't think that it's good. You can see, is it a SNV? Is it indels or structural variations? It's quite complicated to do this. And so we improved the visualization of the variations. And yes, one of them, so when we, first of all, we just read the annotation from the VCF, because VCF 4.2 can store a structural variation. And we, first of all, display it as is from VCF. So if this is annotated as a breakpoint, you will see that this is a breakpoint and a small thin line with arrow to location. So if you have, for example, breakpoint connection between two chromosomes, so all chromosome, you will see the line and connection to the chromosome and position where this breakpoint leads. If this is, for example, inversion, also you will see this as a long inversion covering the whole region, and you will see the link to the next end of the inversion. And this is just the information from the VCF. What we did from scratch, it's we're trying to visualize the rearrangement. So if, for example, we have the variation on genome with annotated genes, we can go to, I don't know, to NCBI or to Ensemble. We download exon structure and visualize the and visualize the result of this variation from the exons level. We did it because we had an example a part of oncogenic variations. So, for example, inversion between, I believe, ALK and EML4. It's a one well-known pro-oncogenic inversion. And for example, for this inversion, we just collect the exons from the first protein. We collect exons from the second protein. We make this inversion and combine two hybrid proteins and show it.
Yeah, thank
I'm
you.
not sure.
Yeah.
I probably can provide the screenshots. I'm unfortunately not at my computer right now, and I don't have the NGB installed. Probably yes.
Okay.
This is one you can.
Yeah, this
you
is the
can go to
repository. Is there any like
Yeah.
instance which is an example one or not? I don't.
It should be. I don't know whether it is active for now because it was not maintained, I believe, since 2018 probably. But you should have here the examples. Here is yes. Let me.
I think this is the example of alignment. It's not the example.
So this is
okay.
This is example of the variations by the way.
Yeah, by
you can download and run it from Docker, but the Docker with example files takes about 12 gigabytes. So, for example, this is rest of the inversion. So you can clearly see the reads which with the mismatched orientation, and you can see this point with the double arrow. So it's cause a variation, and the line which goes to the other side of the variations which is displayed in a separate window. And so by click, I don't know, but probably on the same page you will see how the fused variation looks. Yes, I forgot to install the. So it probably should be somewhere here.
I think that
do you have any like a screenshot or something like this?
I think in here there are no more screenshots except for this. Again, this is the markdown of the repository, which is just googled by like new genome browser. Yeah, I'm just showing it.
So I probably know where I can do it.
Okay.
No, it should be here. It's a README. In README, probably it should be separate link to documentation. Yeah, latest.
Okay.
Probably yes. Latest HTML is no possible. So Markdown documentation.
Markdown documentation. Okay, let's try to do that. Yeah, because I remember this. Yeah, I also had my instance somewhere, but I didn't have. I don't have
user interface. So variations plus where it should be. It should be a variant. It should probably in the variants.
Let's
not,
see. Yeah.
I'm not sure.
Okay, yeah, this is a small variance.
That's how yeah, here.
Okay,
no,
yeah.
This is here it is.
Yeah.
This is the picture that we're doing. In fact, it's not rocket science. It just correctly fetch the data from for example, NCBI, then correctly manage these exons and display the protein with exon structure. So, for example, here you can see first it's initial place where the inversion, and you can see this ALK and the EML4 genes, and so the system it displays you the line, so the point of the inversion. So you can see one part of the inversion between IML, and second one probably at the right point. Could you scroll screen a bit to the right?
To the right. Okay, let me.
I don't know.
Maybe I can just.
I don't see the second. This is the issue of my mobile one. I can
probably yeah, because I have like the.
So here it is, and you can see this is the ALK gene, which then goes to the opposite direction. So you read the EML4 and then go to the back ALK, and this is the first line. And at the second line, you can see the two fused genes between ALK and the EML4, one and the second fused gene. So that's how it looks like, and also we have the same visualization for SNPs and for short indels, but this is not so important. This one, it's the for structural variation separately, and this is the paper quality images which you can insert and stuff. And it's so there is no some I think that's a very good thing, and I like it how we did it in past. But there are no any complicated algorithms. We just extract from the VCF all data from structure variant calling, use some fetching data from database, and good visualization. That's it.
Yeah, thank you for the reminder. Yeah, because I as I've said, yeah, back in I think so like 5 years ago probably I had my own instance to show, but I don't have it now with me. Yeah, no, that's nice that we found these images because I remember, yeah, that was one of the features of the still.
Yeah.
Let me maybe ask you. So, German, what do you think? Like, is there any maybe other alternatives to these kinds of visualization, and whether you consider that this is like it might be easier to interpret in like if you look at these kinds of images or not? I'm asking because, well, that's probably can be seen as somewhat like a direction of research if we can just like extract and maybe do like a separate tool which does these kinds of images. Yeah, whether you consider that that a good approach, or do you have maybe any like?
No, the visualization is definitely good. The visualization is pretty cool. And yeah, maybe that this is not a frequent scenario, this fusion gene, where like
from our customers, from AstraZeneca, work with the structure.
yeah,
Use this
of course it may happen. The
visualization, but
I think gonna have some connection issues. Yeah, probably.
Okay. Yeah. So yeah, it's a very cool visualization, and there are examples of genes which happen exactly. Indeed, yes, in cancer it's pretty frequent. Fusions in cancer are frequent, but in the rare disease, not so. And here it looked good. Like in IGV, you can use this split screen to see both breakpoints there. So technically, you kind of can do this, but you need to press some buttons to do. So I like this solution. I cannot really criticize it. It's just like in the end, the person who interprets the data has to merge this in their head, I would say. So this visualization is kind of, it's not universal. And for most of the cases, it wouldn't be that helpful. It's just like, I don't know, one end of the gene is gone. What happens with the gene? Who knows? Yeah. So it's cool. Yeah. It's not a criticizing, it's
Yeah, thank you for the comment. Yeah, I guess I really, yeah, when I first seen it, I also like thought that it might be a useful thing, and that's why actually I asked. Yeah, and thanks to Gennady for joining us. Yeah, and reminding us about this thing. Yeah, so this is, I think, the important issue of like visualization interpretation. One more question which I wanted to ask you. I think,
of course, we took in coverage and all. For example, the split screen mode. It's also from the IGV directly. The only thing that we did is the visualizations and navigation. We added some variation table where you can just click on the variation and go to it in the coverage view. That's what the technical and UX optimization. So no complicated algorithms.
Yeah, again, thank you again for commenting on that. Yeah, and yeah, the question which I wanted to ask German about the interpretation. So in terms of again the short variants, there are guidelines which we all know, the ACMG guidelines, yeah, as the main ones. Is there any kind of like standardized guidelines for CNVs and SVs or not in terms of like classifying them, or is it just like if it affects the gene or if it disrupts the gene, then it's pathogenic and otherwise not? Are there any like rules for maybe a more stratified approach in terms of like pathogenic, likely pathogenic classification?
There are, it's CNV. They developed it, but it's not. No, it's not the ones that you apply for SNVs. It's not this PP1, PP2. No. It's the quite complicated calculator. You have to put all the data in there, and then the score will be estimated for this variant. So in practice, it's not so convenient. So I guess people still work mainly with the general ideas, like how the gene is disrupted. So there are tools that calculate this CMG calculator for you, but still not widely spread practice here. But indeed, there are criteria.
Okay, yeah, that's it's good to know because somehow I've never come across these. Yeah, and maybe one of the final questions from my side is the issue of validation because we discussed last time with short variants, the idea of like the necessity of validating the alternative methods, and we've discussed that in some centers this is like mandatory, and in some cases, as in your case, yeah, you've said that you do not validate short variants generally by Sanger sequencing. But what is the recommended way of validating CNVs and SVs? Because this is more complicated, I think, in terms of like naturally you can do the PCR or qPCR, I assume, but I don't know what are the guidelines. So if you can share a little bit about this part of the workflow.
If you know the breakpoints, then the PCR would be the easiest way. If the mappability is good on the parts, because you can get just non-specific product there. And it's pretty frequent in short-read genome that the ends of the CNV are in unmappable regions, and you cannot really do PCR there. Because, yeah, you can design primers, but they would not work as you expected. So even for these complicated genes, there are somehow MLPA probes invented. So MLPA with SMA somehow works well, even though I really don't understand why it works well. Yes, but it's still the probe works. What else? In the beginning, the gold standard was to validate everything with arrays. So whatever you find is large CNV, you will do with arrays. But then people realize that NGS is maybe nowadays with the modern tools, modern approaches are even more accurate than arrays in typical arrays, of course, not these super high definition ones, but so it doesn't make really much sense. So anything which is goes in parallel, if you doubt the variant. If the variant is huge and you see multiple types of signal, paired end, split read, and read depth, if the signals combine and the phenotype match and the inheritance match, then you don't really, I would say in modern clinics, you don't validate these things. It's called clear-cut variant, and there are criteria how to define this clear-cut variant. But otherwise, indeed, many approaches depends on the region, I would say, and the information if you
Okay, and is the RNA sequencing typical thing or not? You mentioned that you can do this naturally for certain types of variants, but like how common is it to apply this to us?
So which sequencing? Long read?
RNA sequencing. No, I mean the testing for the expression. Yeah.
RNA sequencing comes into clinic definitely. So it's not solving all the problems. Many genes are not expressed in blood simply, or in fibroblast, whatever you can get without pain. So many patients have good structure event, for example, and they don't have diagnosis because for that you need to take biopsy of the brain, for example, and not many live patients really agree for this biopsy. But yeah, in postmortem, you need to do it really fast, if I understand correctly. So yeah, there are still many mysteries which will never be solved, even at all the technology levels. Model organisms can help, of course, there. Yeah, but it's general problem of medical genetics. It's not structure variant specific problem. Yeah, it's for everything.
Okay, yeah, thank you so much. Yeah, that's it for my questions. Yeah, I think there are a couple of nice questions that I've seen in the chat. So I think one was about the privacy issues. So naturally, we all know that you cannot share genotypes of short variants because these are like, of course, very depersonalizing in some sense, yeah. And is this like the question from the chat is whether it's true for CNVs, like is the CNV profile the same way like the personal personal kind of information which you cannot share the same as S&P genotypes. Again, my impression is that probably that should be the same, but maybe you can comment on that. Are there any differences in treating these different types of variants in terms of privacy?
Yeah, it's exactly the same, but the most of the tools, like maybe 99% of them, are based on the short variants. So it's you have to probably develop your own if you want. But it's possible to identify the person. It's just like most of the CNV calls results will have a lot of false positive, and it will obscure the identification. But if you bother enough, then you may identify patients with CNVs too. Just like no one bothers.
Yeah, and I think one more questions which I've seen somewhere in the chat was about the LLMs, which are of course a very like high point now. Yeah, a lot of talk about all sorts of AI in there. Maybe you can like more broadly comment on if you think that AI may come here like in this field of CNV and SV detection. Not specifically large language models, but the other types of AI. Are there any like prospects for that?
In my dreams, AI would definitely come. For example, currently to interpret the variants, I need to look with my own eyes at the IGV, and I really think that deep learning models can replace me. But then I went to do this deep variant paper and realized that even for short variants, they required huge data set, and the size of the picture that they worked with was quite small. And for structure event, you really need to fit a lot of information there about neighboring reads. So it's a good idea, and I wish this to be implemented, but I doubt it's possibility that it's possible to see it in the recent future. So you need to have a huge database for this. It would be nice. It would improve the interpretability of the calls, but yeah, I don't see huge progress in this field. I don't expect also. There was one more question, I think, from Ivan Balashov. It's like there are sources of variability, which is GC content and mobility. Are there any important sources of coverage biases? Of course, different exome enrichment kits, different wet lab procedure. If you use this protocol from none of volume of DNA, it affects even though the rest of the wet lab remains the same. PCR, if you use extensively PCR, it will really it may destroy your data at all. Like if you have really small amount of DNA and you could amplify it really up to the degree, you know, this is like how it's called, the branching process. When everything is increasing 2 times, then the disparity in coverages increases with each cycle of PCR you make. So indeed there are many other biases, and the only good way to avoid them is to use the control cohort, which is processed with exactly the same protocol as you used right now. Otherwise, trying to include them into your model, like mathematical model formula, you would not succeed here. And the last question, it's what algorithm of normalization using CNV? I think it was something very very easy. I just choose median for each region. I choose median of coverage and say if it's deployed, then median shows the deployed coverage, and the multi-allelic variants they process separately with a separate algorithm within clean CNV with the same segmentation algorithm, but the different normalization procedure. So for rare variants, you just choose median of coverages there. That's of course it allows for GC content, for length of probe. That's actually very interesting source of bias, because for example, Agilent, they used hybridization probes of length 120 base pairs, and if the Agilent is 121 base pair, they must use 2 hybridization probes to cover it, and the coverage is 2x actually there. So you need to take this also into account. Yeah, so basically these are main things. Yeah, if you have a cohort large enough, you may run some sophisticated methods like some similar to these combat methods, which remove batch effects from RNA-seq. You remove principal components, so you use same tricks there. So if you have large cohort, you have power. Yes,
Yeah, I think there also was one question which we've discussed with you before we kicked off the meeting, which we've not covered in this discussion. So maybe if you say a few words about mobile element insertions, I remember that you were mentioning that that's a topic worth talking about. Yeah, so maybe if you have something to add about the specifics of identifying mobile element insertions.
Yeah, surprisingly, structure variant tools, which should detect insertions of novel material in the genome, they don't work well with detection of mobile elements. I guess just detection of insertion is difficult there because there's not so many paired end information. There are split reads that you can see, but not actually paired ends there. So you have to run special tools which are like melt or scramble for the whole exome sequencing. I would recommend to use scramble. It's fast and it detects almost everything. And for genomes is melt. It's rarely causal, but when it is causal, it's very important to know. The most famous case, I don't know if you heard about this, it's the case of the girl who had the personalized drug developed for her. The girl was called Mila, and the drug was called Milacen. And when they performed the testing, they found the point mutation in one allele, but they were missing the second mutation. It was intronic mobile element insertion there, and they were able to uncover the whole story using the methods for detection of mobile elements, but they lost time. And unfortunately, this drug worked for several years, but it was applied too late. So unfortunately, this story didn't end up as the absolute happy end. But we have to keep in mind that these mobile element insertions, they exist and they may cause problems in patients. Super rare, less than 1% in whole diagnostic volume, less than 0.1%. But for unfortunate people who have these mobile elements, it's important to detect them. So yeah, up to you if you want to include this or not, but I think it's ethical to include because structural variant callers often miss these mobile elements. And there are questions. What threshold of likelihood from CNV would you recommend to use for CNV filtering analysis? It depends on the use case. If you use shallow genome sequencing, it's one threshold. Exome sequencing is 100x, another threshold. And the genome sequencing is 30x, third threshold. But I would say like this log likelihood, it's the how to say, it's a ratio between the ratio is the likelihood of two models. So more than 30 for most of the use cases, it probably would work. And the last question is like, how many such Milas exist undiagnosed? That's a very good question, but it's unfortunately it's a topic for we can make a separate seminar for undiagnosed genetic disease. I think it would be very interesting, and it's another topic I specialized in here. Tons of them, a lot of them, and for example, in next June, this June, I'm sorry, in 20 days, there will be a hackathon for solving undiagnosed patients, and there will maybe like 20 families there, which will be profiled with all different technologies, long reads that we discussed today, the optical genome mapping and RNA-seq and short reads, everything, and even this doesn't guarantee that the patients will be diagnosed, unfortunately. With all our knowledge, still, yeah. So how many exist undiagnosed? Millions, really, hundreds of millions probably. Because rare disease is called rare because this particular disease is rare. But if you take all the rare diseases in the world, it will be quite frequent. It's like 1 out of 10 people will have rare disease, and half of them remain undiagnosed. At least half.
Yeah, you've just mentioned. Yeah, let's do that. I think let's do a separate seminar on undiagnosed cases. I think we should do this somewhere in after
Sure.
the topics which I've announced. But yeah, let's do this discussion. I think that would be very nice. Yeah, to talk about that because there is a plenty of subject matter in this field. Yeah, okay, I think we have to wrap up our discussion with that. Yeah, thank you very much for a very, very like nice and detailed information about the field. I hope that was informative and useful for everyone who has listened. Thanks to everyone for joining us. And yeah, we'll see you next time when we discuss somatic variation. Yeah, thank you. Thanks to German, and thanks to the audience.
Ciao.
Yeah, goodbye, everyone.